Welcome, Fred, to the Collaborative Research Center, Origin and Function of Meta-Organisms. It's great that you're here. You are one of the founders or the members of the founding generation of microbiome research, and everybody in the field knows one or two of your very early papers, which discovered, as you say in the title of that paper, the gut microbiome is a major environmental factor and heavily affects the phenotype of a mouse and most likely also of the humans. So before we start a little bit to talk about your science, how did you get to that? You choose a postdoc position, but did you know what you will expect there and what you get there? How did you get to that, and why did you find that interesting?
First of all, thank you very much for having me here. And I did my PhD thesis with Agneta Richter-Dahlfors and Staffan Normark on why do we have E. coli in the gut and they're commensal, and we get the same bacteria into the urinary tract and we get disease. So we must perceive bacteria differently. And that really put the focus for me on the normal gut microbes, which was sort of unfashionable to study in the beginning of 2000. So I was lucky enough to know people that had been working with Jeff Gordon in the past, and I was very impressed by his work. So I took a shot and sent him an email and asked a year and a half before I was graduating if I could come to his lab and do a postdoc. And somehow he accepted me, and we started to discuss what I should do. And I told him that I wanted to work on communication between the microbes to the gut and to the host in the realm of metabolism. First, I wanted to work on the nervous system, but he said I was not skilled enough for that, and he was probably right. Then I said, okay, let's work on enteroendocrine cells. But then we said that, okay, before we do that, we should probably do some more basic experiments first. And that was to see whether there was a difference in amount of body fat between a germ-free and a colonized mouse. And that sort of started my career in this field. Because what we found was that there was much more body fat in a colonized mouse. And we could take a germ-free mouse and colonize it, and by adding back bacteria, we have more fat, although the mice would eat 30% less. So at that time, we then start to think that the microbes are helping us digesting food components that we cannot digest ourselves. And on top of that, we also soon thereafter found that it's probably also controlling the metabolism in the host, potentially via nerves or via hormones. And that's a lot of things we have to learn still about that.
So the early days and this early papers made very strong messages, which were very good because they alerted the field that there is something interesting in this not important beneficial microbes. The strong messages were maybe a little too strong, and probably you will take a little back from that at the early days, but I still think this were just absolutely important papers. Now,
yeah, I think that, just if I can interrupt you there, that I think that the data is extremely reproducible. It's I think that we cannot really translate it into humans. I think this is a mouse phenotype, as we as humans will always have bacteria, and having a completely sterile gut is nothing we see in humans. So you're absolutely right that we you're a little bit like a bit overambitious, and then you sort of settle down. But it's very rewarding to see that how this field has come together and start to find mechanisms and causalities that we didn't know just 10, 15 years ago even existed.
Yeah, absolutely. And I think this is the important message that the field is exploding. Many people are joining that now, and but we have really to stay to causality, and correlation doesn't mean too much. You have to start with description, but then you have to move on to some causality experiments. Now you are heading a big laboratory in Gothenburg and in the Wallenberg laboratory, and you compare the time in 2003, 2004 to today. Of course, our approach to things and to seeing something invisible, which we do every day, is heavily dependent on technology. And technology has changed a lot. And you have all the state-of-art technology to your availability. You may say a few words. I mean, how did technology and modern equipment and not only sequencing, visualization and stuff like that, that must have had a tremendous influence in what you now see today.
Yeah, I think especially if you look at the sequencing and the enumeration of bacteria. When we started in 2003, we did full-length clones, and we picked every colony and we sequenced by Sanger sequencing. And the biggest study that was done was in Jeff's lab with 20,000 clones. And today, people would laugh if you have 20,000 reads, maybe in one sample, it's okay. So I think that the limiting factor has really shifted as technology has moved really fast. And to beginning with was the sequencing that was a limiting factor, because you did the bioinformatics in an Excel sheet. Then when we moved to the barcoded pyrosequencing, well, then the limiting factor was no longer the sequencing cost, it was rather the data handling and the programs. And I think now we're at a point when it's coming more to the design of the study, that we don't get better data than we can control for.
Does it mean that today we don't have limits anymore in seeing this invisible world?
I think
where would be limits?
I think that we still getting we have a good picture on the most abundant bacteria, but the lower abundant bacteria we know less of. And I think this is a conceptual important issue. If you take, for example, Clostridium scindens, which is the bacteria producing secondary bile acids such as deoxycholic acid, it's quite often you don't find it in a metagenomic analysis. But if you do a qPCR, you find it, and you definitely find deoxycholic acid. So some of the minor components may have extremely important physiological features. So understanding also moving from these broad descriptions we have had in the past, like genera, I think we have to come down to strains. It's some of the lactobacilli probiotics, for example, if you just differ by a few nucleotides, you may have the capacity to produce histamine from histidine or not.
Yeah. I fully agree, and actually I got this message early on by a former guest of ours, and Eugen Rosenberg. When you come from a more ecological and evolutionary adaptation point of view, Eugen always said, the rare bacteria may be the most important ones, because they may colonize a tissue at some point, and then they may give the whole microbiome a different touch, but sequence-wise we may still miss them. Yeah. You said a few minutes ago already the mouse is the mouse, and certainly the mouse is not a human. Is the mouse a good system then?
This is the question we all are sort of fighting with. I think we have to break it down to a few points. First of all, we need to have a model system to do this analysis, because doing it in a test tube or in a cell culture dish is impossible, because you need all the organ systems present. I think that the mouse is probably not the perfect one, but there's a lot of advantages that I think weighs pretty strongly, and that is that we know we can do genetics in the mouse relatively easily. It is the food it eats you can make it similar to the humans, because I think that the food is an important component. That said, what we will miss is, for example, there's nice work from several labs showing that segmented filamentous bacteria, for example, are very host-specific. So if you're studying them, a mouse may not be the right model organism. So I think if we just trying to use it as a model and not as a truth, which I think scientists will sometimes make something in mice and say this is true in humans. If we start by looking at phenotypes and observations in a human, and we try to model that in a mouse. I think we're better off than trying to first find something in a mouse and then see if it's true in a human. What's striking in comparing human and mice, or human and mouse microbiota, for example, is that it seems like the human microbes can induce a lot of the metabolic functions in the gut, if you look at transcriptome, but not the immune functions. Mm-hmm. So once again, it's important to know what question you ask, yes, to see if you can, yes, so you don't miss anything or overinterpret something else.
Yes. So getting conceptual insights, it's very good, but you have to know you are in the mouse. On the other side, the medical doctor who says, I'm only interested in the human, I mean, please then tell me how you want to find causality in a human system.
Yeah, exactly. So what I usually tell is that in humans we find importance or relevance, and in mice we can find mechanisms. But it's that,
absolutely.
But it's otherwise we need both.
Oh yes, and I mean working with a unimportant invertebrate, but of course the human is always in my mind at the end. Okay, but to understand fundamental principles, we need models. That's very clear. If you take your studies comparing germ-free versus conventionalized or control mice, the early days, you took a number of animals, a number of specimens, and then you came up with a nice figure, and it has a standard deviation, and there's some variation in between, and then you made the message, and then you made a conclusion out of that. Novel technology, and nowadays we know that that average number and any conclusion from that average number to a given individual in a given population in a given environment may not be a good idea, because everybody seems to be different. Personalized medicine, precision medicine, that's the new word. What do you think about that?
I think it's extremely important in the field of microbiome, because there's a number of things I think we need to clarify, and that is people start to talk about the healthy microbiome, but I think that we know too little about what health is.
Do we know the healthy microbiome?
No.
Yes, no. Yes.
So I think we can say the normal population microbiome, or the normal variation in microbiome. And then if you start to think about the diseases that we most often framing can have several different etiologies, several different causes with the same eventual output, the same disease markers. So I think that in the case of diabetes that we work with is that you can have diabetes of many reasons, and maybe some of them is caused by our genes, others by what we eat, others by bacteria perhaps, and others by an interaction between these three, or viruses for that matter, or fungi that you like. That said, I think that then it may be even further stratification that's required, and that for given phenotypes there may be a subset that has a microbial component. And by stratifying to subgroups, I think to go to individuals will be too hard today. I think we may have a much better possibility to help those individuals. And I think the clearest result so far is from Aaron Segal and Aaron Elenov's dietary interventions, when they really can do much more personalized dietary advices based on the microbiome. So I think that the future is there, and I think that's partly also why we have to leave the old probiotic concept, where it was give a living bacteria to promote health, to a more drug-based approach, where you treat the bacteria as a drug, and we have to do placebo-controlled, double-blinded studies, and verify them in independent studies, like we do for any drug, before we can make the claims we would like to make. And they probably would not be as strong as what we would have hoped 10 years ago.
Yeah. We come back to the probiotic and maybe the prospect for the future in a minute. Before we do that, your approach or your suggestion of stratifying a given population is, I think, very elegant, because it makes the whole personalized medicine approach feasible, because I do not think that we can really individually consider thousands, 100,000, millions of people, but if you put them, and that's I understand how you propose to do it, into subgroups which you characterize, that makes it kind of feasible to more specifically address a given group of persons. Is that how you propose to go on?
Exactly. And so in the case of diabetes, we generally talk about type 1 and type 2. And I think we have to say maybe type 2A, B, C, D, E, F, G. And then a couple of those letters may be tractable for microbiome, others not. And then maybe there's different microbes for these different letters.
Yes, yeah. So going to the potential of treating diseases or preventing, even better, preventing diseases, which is not possible these days yet. Of course, probiotics were for nearly 100 years now. There's companies around and they sell things and people believe that this is good. Nobody knows what they are really doing and how they work. But of course, your research and research from other laboratories contribute to a very clear understanding that there is also an industrial aspect to that. There's an aspect of startup companies to that. There will be some commercial value behind this basic research. Would you agree, and do you see that in this way?
Absolutely. I think it's finding the golden bullet, so to say, the bacteria that would have a role will be what tons of startup companies are doing, and but before, I think that there's a number of challenges that has to be overcome before we can do this. I think it's first of all there will be more prescription type. I envision that you take a given probiotic for a given disease or a given phenotype. But many of these may be anaerobic bacteria, so they would die in oxygen. So I think that it's not as simple as just taking a bacteria that you find and then say it will be a product. I also think that these are likely organisms that will not be under the grass, so generally regarded as safe. So you have to do safety trials. So we have to start looking at it more like, as I said before, like from the pharmaceutical industry, and also how should this be handled by? Is it so a little bit on that? Is it from the FDA or the EFSA or the pharmaceutical legislation? Should this be considered a drug or a food? And I think that's still also a lot of discussion there. But to my mind, I think we have to move to much more research-based products rather than let's find another lactobacilli and see if it works.
Yeah, bringing that back in the context of our center, meta-organisms, where we think that we have to understand the mechanisms, how interactions work within this multi-organismic association. When I visit occasionally probiotic or pharmaceutical companies, and then I get shining eyes why they have found the bacterium which does the thing. And so the evidence always is then you take this bacterium or this microbe on a plate and you culture it, and then you do all kind of enzyme assays and things, and you find out what this wonderful thing is doing, and then you claim, and then you say, well, this is producing this and this, and that interferes with your metabolism. What we learn in the last few years is, of course, that the performance of a microbe is highly context dependent, and it never will do the same thing on a plate as it would do in your body. So that's the challenge, of course, and that brings back the importance of model systems, where you at least have to give some level of complexity which you can study, and but then you get a real answer, or hope you get a real answer. And this simplistic view of look to a microbe and do simple biochemical readouts, that's probably too simple.
Yeah, absolutely. And I think that what you said is also highlighted in several studies, including the fecal microbiota transfer experiments, that you will end up with responders and non-responders. And why some respond and some do not could very well be the background microbiome, and you will never pick that up on a plate. And understanding the interaction between bacteria and between bacteria and the host, and bacteria and host and diet, will be very, very important. We have had one study where we looked at Prevotella copri and its capacity to mediate beneficial effects on glucose metabolism, and that turned it in our hands to be dependent on whether the mice had fiber or not. So that was really, without the fiber, they didn't do anything.
Exactly. Yeah. So context dependent. Yeah. Looking a little bit in your large team you work with, and you are so productive. I mean, it seems that every single member is a specialist, an expert, and so they come from many different backgrounds, interdisciplinary working. That's also a key word of how modern research is really done. If I think on our junior investigators and junior researchers, I mean, we have to teach them interdisciplinary working and understanding different languages. What do you expect from a young person when the young person addresses you like you addressed Jeff Gordon many, many years ago, and they say, Professor Bäckhed, can I come to you? What type of training or background or personality, what do you expect from a young person?
I expect a solid basic training in at least one or two fields, like biochemistry or microbiology or bioinformatics. So you have something to sort of fall back on. Then I expect you to come to me because you want to learn something new. So having a natural sense for curiosity is something very important in my mind. That, and then that you have to speak decent English at least, because I think communication is essential. And above all, to being a kind person and having integrity. So what we do in the lab is that if you want to join the lab and if you pass the first initial screening, you will come for a 2-day interview and meet everybody in the group. And then we decide as a group whether that individual is welcome or not. Because I think that in science, usually it's tough experiments. And if you fail a couple of experiments, you need to feel safe, cared for in the group, and like to be at work. So you can persevere during the hard times, and at the same time can share the joy of success with others without feeling envy or jealousy. So I'm trying to really have this team spirit, and that we should be one big family, criticize each other when we have the doors closed. If we open the door, we should support each other, and always kind of feel safe. I think that's very important.
Mm-hmm. I think that's a wonderful way of how to establish a creative group. What I tell my people always is or outside visitors, what we are doing is we are not producing something really, not a product, but we produce ideas. And to produce an idea, it needs an atmosphere. And the most important thing which you gave us in your evening talk tonight, and which we share, I guess, is even if we approach age, we still have fun. And I want that the young people also have fun in what they are doing. And I think if one realizes that some person is suffering, or that's not a good idea to spend then some time in a scientific lab.
No. It's like I generally tell people that we're very fortunate. We are like football professionals, that we can work with our hobby. Yes. And I think if you don't feel the same, you're competing with everybody else that live and breathe science. So you cannot be successful. And also that I generally tell people that if you come to me as a student or a postdoc, I will try to give you the best situation to do good research. But then by the end of the day, it's up to you. So you get freedom. You can be at work more or less whenever you want. But don't come to me and complain that you didn't make it. I try to give everybody the same chance.
Yeah. I think that's most important points and key to success. Thank you very much, Fred, for coming and sharing some time with us.
Thank you very much.
Thank you.